Chapter 1. The Digestive State. Digestion begins as a state, not as a reaction to food. Before any mechanical breakdown or chemical transformation occurs, the body must enter a mode that permits those processes to operate. This state is governed by the nervous system, specifically by the balance between parasympathetic and sympathetic activity. When parasympathetic signaling is dominant, the body allocates resources towards secretion, motility, and coordinated digestive output. Blood flow is directed toward the gastrointestinal tract, glands increase production of digestive fluids, and the sequence of events required for processing food is allowed to initiate. When sympathetic signaling dominates, those same processes are suppressed. Blood flow is redirected towards skeletal muscle, secretion is reduced, and motility becomes less coordinated. The system does not partially engage digestion under these conditions. It restricts it. This state acts as a gatekeeper. Food entering the system does not automatically trigger full digestive function. The nervous system determines whether the system will operate at sufficient capacity to process that input. If the body is in a constrained state, the early stages of digestion are already compromised before food reaches the stomach. Salivary output may be reduced, sensory signaling is diminished, and the anticipatory phase that prepares the stomach and pancreas is weakened. By the time food arrives in the stomach, the system is already operating below its required output, and the sequence begins with a deficit. Gastric function is particularly sensitive to this state. Acid production is not constant. It is modulated by neural input that reflects the body's readiness to digest. When parasympathetic tone is present, acid secretion increases to levels that can alter protein structure effectively. When sympathetic tone dominates, acid output is reduced. Proteins enter the stomach but are not exposed to sufficient acidity to unfold completely. This alters the entire downstream process. Enzymes that depend on unfolded substrates encounter resistance, and the material that moves into the small intestine carries structural integrity that should have been removed. The system is forced to compensate for a failure that occurred before digestion fully began. Motility follows the same pattern. Coordinated movement of food through the digestive tract depends on rhythmic signaling that is supported by the parasympathetic system. When this signaling is disrupted, movement becomes irregular. Food may remain in the stomach longer than intended, increasing the opportunity for fermentation, or it may pass too quickly into the intestine without adequate preparation. In the small intestine, segmentation and peristalsis lose precision, which affects both breakdown and absorption. The system continues to move material forward, but it does so without the timing required to maintain efficiency. This state-dependent behavior explains why digestion can vary significantly even when food intake remains consistent. A meal consumed in a calm, regulated state may be processed with minimal disturbance, while the same meal consumed under stress produces bloating, discomfort, or irregularity. The difference is not in the food. It is in the system's ability to enter a state that supports the sequence of digestion. Without that state, the system begins from a position of reduced capacity and carries that reduction through every subsequent step. Chronic suppression of the digestive state leads to structural changes in how the system operates. When the body repeatedly processes food under constrained conditions, it adapts to that constraint. Secretory output remains low, motility patterns become inconsistent, and the coordination between organs weakens. The system begins to normalize dysfunction. What started as a temporary reduction in capacity becomes a persistent operating mode. Over time, this reduces tolerance to a wider range of foods because the system is no longer capable of executing the sequence at full capacity. Restoring digestion requires restoring the state that permits it. This does not begin with food selection or supplementation. It begins with re-establishing the conditions under which the system is allowed to function. When parasympathetic signaling is restored, secretory output increases, motility becomes more coordinated, and the sequence can begin to operate as designed. Each step regains the ability to prepare the next, and the system moves away from compensation and back toward coordinated processing. The digestive state is not an accessory to digestion. It is the condition that determines whether digestion can occur at all. Nervous system control of digestion. Digestion is directed by the nervous system before it is carried out by organs. The stomach, pancreas, liver, and intestine do not decide independently when to secrete or how strongly to contract. Their activity is driven by neural input that sets the level of output across the entire system. This control is primarily organized through the balance between parasympathetic and sympathetic signaling, with the enteric nervous system acting as a local network that executes instructions and adjusts behavior in real time. When parasympathetic tone is sufficient, secretion increases, motility becomes coordinated, and blood flow is directed toward the gastrointestinal tract. When sympathetic tone dominates, those same processes are reduced or suppressed, and the system shifts away from digestion toward immediate survival priorities. The vagus nerve provides the main parasympathetic pathway that links the brain to the digestive organs. Its activity determines how strongly the stomach secretes acid, how effectively the pancreas releases enzymes, and how consistently the gallbladder contracts to deliver bile. This is not a subtle modulation. Changes in vagal output directly alter the volume and timing of digestive secretions. When vagal signaling is strong, gastric acid reaches levels that unfold protein structure, enzymes are released in synchrony with incoming material, and bile enters the intestine at the moment it is needed for fat emulsification. When vagal signaling is weak, each of these outputs is reduced. The system continues to function, but it does so below the threshold required for complete processing. The enteric nervous system operates within the walls of the gastrointestinal tract and coordinates local activity, but it does not override the global state set by the central nervous system. It organizes peristalsis, segmentation, and local reflexes that move and mix material, but its behavior is constrained by the level of input it receives. If upstream signaling reduces secretion, the enteric network must work with material that is less prepared and more difficult to process. Motility patterns adjust in response to this altered substrate. Movement may slow to allow more time for breakdown, or accelerate to clear material that is not being handled efficiently. These adjustments are compensatory. They do not correct the underlying issue of reduced input from the central control system. Blood flow is also regulated through neurocontrol, and this directly affects digestive capacity. Under parasympathetic dominance, circulation is directed toward the stomach and intestines, supporting secretion, absorption, and tissue maintenance. Under sympathetic dominance, blood is diverted toward skeletal muscle and away from the digestive tract. This reduces the resources available for secretion and limits the ability of the intestinal lining to absorb nutrients efficiently. Even if enzymes and bile are present, reduced perfusion constrains how effectively the system can use them. The physical environment required for digestion is altered at the level of circulation. This control system explains why digestion can change rapidly without any alteration in diet. A shift in nervous system state can increase or decrease acid production, enzyme release, bile flow, and motility within a short time frame. The same meal can be processed efficiently or poorly depending on whether the system is receiving signals that permit full operation. When neurocontrol is consistently biased toward suppression, the system adapts to a lower baseline of activity. Secretory glands reduce output, motility patterns become irregular, and coordination between organs weakens. The system is still active, but it operates with reduced precision and lower capacity. Restoring digestion requires restoring the signaling that drives it. The organs involved in digestion respond to instruction. When parasympathetic signaling increases, secretion rises, timing improves, and the sequence of digestion begins to reassemble into a coordinated process. The enteric nervous system can then operate on material that has been properly prepared, and motility stabilizes because it no longer needs to compensate for upstream inefficiency. The control layer determines the performance of the entire system, and without correcting it, downstream interventions remain limited by the signals that continue to restrict output. Parasympathetic dominance and digestive permission. Digestion requires permission at the level of the nervous system before it can proceed at full capacity. This permission is granted through parasympathetic dominance, which shifts the body into a mode where resources are allocated toward processing, absorption, and internal maintenance. Without this shift, the digestive system does not fully engage. It operates in a restricted state, producing lower levels of acid, enzymes, and bile, while motility becomes less coordinated and blood flow is directed away from the gastrointestinal tract. The system is active, but it is not operating with the output required to complete the sequence of digestion. Parasympathetic signaling initiates the preparatory phase that allows digestion to begin before food is even swallowed. Sensory input from sight, smell, and taste activates neural pathways that increase salivary secretion and prime the stomach for acid production. This early signaling sets the baseline for everything that follows. When parasympathetic tone is strong, gastric glands are activated in advance, and the stomach is ready to alter protein structure as soon as food arrives. When this signaling is weak or absent, the system starts behind, and the stomach must attempt to generate sufficient output after the fact. This delay reduces the effectiveness of the initial transformation and alters the quality of material that moves forward. Gastric acid production is directly tied to this state. Under parasympathetic dominance, acid secretion reaches levels that denature proteins and create an environment where enzymatic activity can proceed efficiently. The pH of the stomach is not a static value, but a controlled condition that depends on neural input. When sympathetic signaling overrides parasympathetic activity, acid production is reduced. Proteins remain partially folded, and the stomach becomes a holding chamber rather than an active processing site. The material that exits the stomach carries structural resistance into the small intestine, increasing the burden on downstream systems. Pancreatic enzyme release and bile flow follow the same pattern. Parasympathetic input coordinates the timing and quantity of these secretions so that they match the arrival of partially processed material from the stomach. Enzymes are delivered in concentrations that can complete the breakdown of macromolecules, and bile is released in a way that allows fats to be emulsified into absorbable structures. When parasympathetic signaling is reduced, these outputs become inconsistent. Enzymes may be released in insufficient amounts or at the wrong time, and bile flow may not align with the presence of dietary fats. The system loses synchronization, and each stage operates with reduced efficiency. Motility is also governed by this permission state. Under parasympathetic dominance, the digestive tract moves material in coordinated patterns that allow for thorough mixing and controlled progression. The stomach empties at a rate that reflects the level of processing achieved and the small intestine segments and propels material in a way that maximizes contact with digestive surfaces. When this state is suppressed, motility becomes irregular. Food may remain in the stomach longer than necessary or pass too quickly into the intestine without adequate preparation. In the small intestine, segmentation loses precision, reducing the effectiveness of both breakdown and absorption. Digestive permission is not an abstract concept. It is a measurable shift in output across the entire system. When parasympathetic signaling is present, secretion increases, timing improves, and the physical environment required for digestion is established. When it is absent, the system operates under constraint, and every stage begins with a deficit that carries forward. The difference between efficient digestion and chronic instability often begins at this level. Without permission, the system cannot perform the transformations required to convert food into usable material. With it, the sequence can proceed with the precision needed to maintain stability. Stress as a suppressor of digestive output. Stress shifts the body into a state where digestion is deprioritized at the level of control, not at the level of food. Sympathetic activation alters the allocation of resources, directing blood flow towards skeletal muscle and away from the gastrointestinal tract, reducing secretory output and disrupting the timing that coordinates each stage of digestion. The system does not shut off completely, but it operates below the threshold required to complete the sequence. Acid production drops, enzyme release becomes inconsistent, bile flow loses synchronization, and motility patterns become irregular. Material continues to move through the system, but it is processed under constrained conditions that favor incomplete transformation. Gastric function is one of the first areas affected. Acid secretion depends on neural input that reflects whether the body is in a state that permits digestion. Under stress, that input is reduced and the stomach produces less acid. Proteins enter but are not fully denatured, which limits enzymatic access later in the process. The stomach retains food longer as it attempts to compensate for reduced chemical breakdown, increasing the likelihood of fermentation and pressure buildup. In some cases, the system accelerates emptying to move inadequately processed material into the intestine, trading preparation for speed. Both patterns originate from the same constraint. The stomach is operating without sufficient output. Pancreatic and biliary responses are also altered. Enzyme secretion depends on coordinated signaling between the stomach, intestine, and nervous system. When stress disrupts that signaling, enzymes may be released in lower quantities or at the wrong time relative to incoming material. Bile release becomes less predictable, which affects fat emulsification and the formation of structures required for absorption. Fats pass through the system in larger, less organized forms, increasing the burden on the intestine and altering the composition of material that reaches the colon. The downstream environment shifts as a result, not because of the food itself, but because of how it was processed. Motility changes under stress in ways that reflect the system's attempt to manage incomplete processing. Peristalsis and segmentation lose coordination, leading to either slowed transit with increased retention or accelerated transit with reduced absorption. These opposing patterns can occur within the same individual depending on how the system responds at a given moment. When transit slows, fermentation becomes more prominent as microbial populations act on substrates that were not fully broken down. When transit accelerates, nutrients pass through before they can be absorbed, leading to variability in energy availability and nutrient status. The system adapts to constraint, but the adaptation introduces new inefficiencies. Circulatory changes reinforce these effects. Reduced blood flow to the digestive organs limits both secretion and absorption. Even when enzymes and bile are present, the tissues responsible for processing and transport operate with fewer resources. The intestinal lining has less capacity to move nutrients across membranes and maintain barrier integrity. Larger fragments are more likely to interact with immune tissue, increasing the probability of inflammatory signaling. These changes are not isolated. They are coordinated responses to a state that deprioritizes digestion at multiple levels simultaneously. Chronic stress extends these patterns into a stable operating mode. When the system repeatedly processes food under constrained conditions, it adapts by lowering baseline output. Secretory capacity declines, timing between organs becomes less precise, and the coordination required for efficient digestion weakens. Symptoms become more consistent because the system is consistently operating below capacity. Food appears to be the trigger, but the underlying condition is a control state that limits the system's ability to perform the necessary transformations. Restoring digestive function requires reversing this suppression at the level of control. As sympathetic dominance decreases and parasympathetic signaling increases, secretion rises, timing improves, and blood flow returns to the gastrointestinal tract. The stomach regains the ability to generate sufficient acid, the pancreas and liver restore coordinated output, and motility stabilizes because material is being properly prepared at each stage. The system no longer needs to compensate for incomplete processing. Digestion becomes more predictable because the conditions required for full transformation are present again. State-dependent digestion variability. Digestion does not produce consistent outcomes from identical inputs because the system that processes those inputs is not constant. The digestive tract operates according to the state imposed by the nervous system, and that state can shift from one meal to the next without any change in diet. Acid output, enzyme release, bowel flow, motility, and blood distribution all respond to this control layer. When the state supports digestion, these processes align in timing and magnitude. When the state constrains digestion, each process operates below capacity or out of sequence. The variability observed in symptoms is a direct reflection of these shifting conditions rather than an inherent unpredictability in the food itself. Gastric performance illustrates this clearly. In a state that permits digestion, the stomach generates acid at levels that unfold protein structure early in the process. The material that exits the stomach is chemically altered and ready for enzymatic reduction. In a constrained state, acid output is lower and proteins remain partially intact. The same meal enters the small intestine in two different forms depending on the state under which it was processed. Enzymes encounter either an accessible substrate or one that resists cleavage, and the efficiency of breakdown diverges immediately. The difference is established before the intestine has an opportunity to act. Pancreatic and biliary responses amplify this divergence. When signaling is intact, enzyme release is synchronized with the arrival of partially processed material, and bile is delivered in a pattern that supports fat emulsification. Lipids are organized into structures that can be transported across the intestinal lining, and the system proceeds with predictable absorption. When the state is constrained, enzyme output may be delayed or insufficient, and bile release may not match the presence of dietary fat. The same fat intake can be efficiently emulsified in one state and poorly organized in another, altering both absorption and the composition of material that continues through the gut. Motility reflects these upstream differences. When material is properly prepared, the digestive tract can move it through in coordinated patterns that maximize mixing and contact with absorptive surfaces. When preparation is incomplete, motility becomes adaptive. The system may slow transit to allow more time for breakdown, increasing retention and fermentation, or accelerate transit to move unstable material through more quickly, reducing absorption. These patterns are not fixed. They shift with the state of the system, which is why an individual can experience alternating symptoms without any meaningful change in diet. This variability creates the impression that certain foods are inconsistent or that reactions are unpredictable. In reality, the variability is structured. It follows the condition of the system at the time the food is processed. A meal consumed under conditions that support digestion is likely to be handled with minimal disturbance. The same meal consumed under conditions that suppress digestion produces a different outcome because the sequence begins from a reduced baseline. The input is constant, but the system is not. Understanding digestion as state-dependent removes the need to attribute variability to the food. It shifts attention to the conditions under which the system is operating. When those conditions are stabilized, digestive output becomes more consistent. Acid production, enzyme release, bile flow, and motility begin to align with the demands of incoming material, and the system produces predictable results from the same inputs. Variability decreases, not because the diet has been simplified, but because the system processing that diet is no longer fluctuating between states that support or suppress its function. Why food quality cannot override suppressed physiology. Food quality determines the potential value of an input, but it does not determine whether that value is realized. The digestive system must convert structure into absorbable components before any nutrient can be used. When physiology is suppressed, that conversion is incomplete. High-quality inputs enter the system with intact structure, and if the machinery that alters that structure is operating below capacity, the outcome is constrained regardless of the starting material. The system cannot extract what it cannot process. Suppression begins at the level of control and propagates through every stage of digestion. Reduced parasympathetic signaling lowers gastric acid output, which limits the unfolding of protein and the activation of enzymes that depend on an acidic environment. Pancreatic secretion becomes inconsistent, reducing the availability of proteases, lipases, and amylases at the moment they are needed. Bile release loses coordination with fat intake, leaving lipids in larger, less organized forms that resist absorption. Blood flow to the gastrointestinal tract decreases, limiting both secretion and the capacity of intestinal lining to transport nutrients. These changes alter the system's ability to perform the transformations required to turn food into usable material. Under these conditions, improving food quality changes the input, but not the processing environment. A nutrient-dense meal still requires acid to denature proteins, enzymes to cleave macromolecules, and bile to emulsify fats. If those steps are incomplete, the material moves forward in a partially processed state. Larger fragments persist in the intestinal lumen, altering fluid balance and motility, and increasing the availability of substrates for microbial fermentation. The system is then forced to manage the consequences of incomplete breakdown rather than benefiting from the potential value of the food. The difference between high-quality and low-quality inputs becomes less relevant when both are processed under the same constraint. This explains why individuals can adopt increasingly refined diets without resolving symptoms. Removing processed foods or selecting nutrient-dense options can reduce certain burdens, but it does not restore the outputs required for full digestion. The system remains limited by its capacity to generate acid, enzymes, and bile in the correct sequence. As long as that limitation persists, the range of foods that can be processed without disturbance remains narrow. The appearance of improvement reflects reduced demand on the system, not an increase in its functional capability. The constraint also affects absorption and distribution. Even when some breakdown occurs, reduced blood flow and altered membrane transport limit how effectively nutrients move into circulation. Lipids that are not properly emulsified fail to form transport structures that can cross the intestinal lining. Amino acids derived from incomplete protein digestion are absorbed less efficiently and may appear in imbalanced patterns. Fat-soluble vitamins depend on bile-mediated transport and are directly affected by disruptions in lipid processing. The system does not selectively fail at one nutrient. It alters the entire flow of material from the lumen into the body. Restoring physiology changes the outcome without requiring perfect inputs. When acid production reaches effective levels, protein structure is altered early and consistently. When enzyme release is synchronized with incoming material, macromolecules are reduced to absorbable units before they reach the distal intestine. When bile flow is coordinated, lipids are organized into structures that can be transported across membranes. Blood flow supports both secretion and absorption, allowing the system to move nutrients into circulation efficiently. Under these conditions, the system can extract value from a broader range of inputs because the transformations required to access that value are being completed. Food quality matters once the system can process it. Without that capacity, quality remains potential rather than realized function. The limiting factor is not what is consumed, but whether the system can perform the sequence required to convert that input into structure and energy.